Hello cell biology fans, here I am with the flipped lecture for today. We're going to quickly go through the chemistry of cell biology. This is all stuff that you learned in 107 and in any kind of chemistry class, but we'll go over it just to make sure everybody's on the same page. The chemistry that drives all of biology is related to how atoms and macromolecular structures are put together, and this is all based on the building blocks of chemistry. And so knowing a little bit of chemistry helps you in biology. You don't need to know tons, but it certainly will get you pretty far in understanding how structure leads to function. And differences in structure are obviously going to lead to differences in function, and the chemistry behind that is pretty simple to understand, so I hope we can get it here. And if you don't understand this, please come and ask me questions. So if we look at science, right, science is based on understanding all the little pieces that get put together, and different sciences, right, study different pieces. So subatomic particles and atoms are all the science of physics, and then atoms and how they're put together, a lot of that is done by chemistry. And then if you look at pretty much, let's see, these four, this is all biology. And who does this top stuff? Humans and other organisms, and cultures and civilizations, sociology, and anthropology. So trying to understand how we put together all the little pieces that leads to cultures and civilizations, and what those cultures and civilizations do to one another, with one another, etc., are different sciences. And we really need to be, as biologists, we need to be a little bit diverse in understanding a lot more of this chain than anybody else. Physics, right? Physics really is just looking at tiny little pieces of the puzzle, without which that information we wouldn't be able to understand all of the stuff that we do. So let's start with understanding water. Since your body is made up of 70% water, right? The earth is made up of 70% water. It turns out it's really important, and life probably wouldn't have evolved on the planet if water hadn't come into being. Back in the olden times, right, water wasn't necessarily here to start with, but as water became available, this was what drove the chemistry behind the first living organism coming into being. So if you look at this little chart, I actually love this little chart because right here, what I'm pointing to is the 70% water, right? You're only made up of 30% chemicals. And what's the number one thing in that 30% chemicals? Proteins. So 15% of that 30%, right, half of the chemicals are proteins. So you're going to start to understand why we really want to understand what protein structure is, how it's affected, how it's changed in understanding biology. Interestingly, your brain is 70% water, your lungs are 90% water, which is a little counterintuitive, right? 90% water in your lungs is perfectly good, 91% and you're drowning, and your blood is 83% water. So if you start looking at this, what happens when you get dehydrated? Well, when you're dehydrated, the number one thing that goes is your brain. You start to get really loopy. You know, you're not really understanding what's going on, and that's because of the physiological effects in your blood, in your lungs, in all of your tissues. So the makeup of who we are, the chemical makeup, is really important. And I said, you know, a significant portion of it is proteins, but if you look at it, right, 1% is DNA, and the rest are all these small molecules, which we're going to talk about, and how they play a role in you being a human. If we look at the elements that are important for biology, all biological beings are made up of four main elements: hydrogen, carbon, oxygen, right? Hydrogen, carbon, oxygen, and nitrogen. They're shown in red on this graph. And then all of the rest of the Earth, right, the rest of the Earth still has hydrogen, carbon, oxygen. As a matter of fact, tons of oxygen as far as the Earth's crust. Very little nitrogen, but calcium, sodium, aluminum, silicon, others. All right, so hydrogen, carbon, oxygen, and nitrogen are the main elements that drive the formation of life on Earth. And it is how these elements get put together in chemical bonds that is really important for how we function as human beings. Right? We talk about organic and inorganic parts of the world. Organic, right, we say, is anything that has carbon in it, right? Organic is carbon-based. And so all of organic chemistry is based on carbon. Inorganic chemistry is based not on carbon. And all the inorganic parts of the world are really what are the non-living parts that we look at. So understanding biology really does mean that you have to understand chemistry and even some physics. So let's start at the very basic, what is an atom, right? So if I say an atom, it's the same thing as an element. And you can find all the atoms on the chemistry chart, the chart of the elements. And every element has a certain atomic number. And what is that atomic number based on? The atomic number is based on, right, the molecular weight of these elements. And what makes up the molecular weight of these elements? So elements are made up of protons, neutrons, and electrons. Protons and neutrons have mass, and electrons have tiny, 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 tiny amounts of mass. So what you're going to see is that the electrons don't account for much of the mass, but they're really, really important for the biological functions of atoms. So here we're looking at two atoms, hydrogen, right? So here's hydrogen, and what you see is it has one proton, and it has one electron. And hydrogen is, right, so the one proton and one protons have a positive charge, the electrons have a negative charge, so if you add those two together, you see hydrogen has no charge. What is its molecular mass? Well, its molecular mass is based on the one proton, and its molecular mass is 1. And molecular mass is measured in Daltons. All of this should be review. I shouldn't really be telling you anything new. But let's look at carbon. So carbon is way more interesting, right? Carbon actually has multiple electron orbitals. So we know that the first electron orbital over here is the first electron orbital, and how many electrons can fit in the first electron orbital? You can have a max of 2. Here's the second electron orbital shown in carbon, and how many max can you have in that? The max is 8. Alright, so let's count the protons. How many protons are in carbon? There's 6 protons in carbon. How many neutrons? 1, 2, 3, 4, 5, 6. 6 neutrons. So 6 plus 6, and how many electrons? 6. Wow, everything's 6. So 6 protons, 6 electrons gives carbon no charge, but 6 plus 6 gives carbon a molecular mass of 12 Daltons. Its atomic number is 6. The atomic number of hydrogen was 1. And if you look on the chart of the elements, it's really easy to follow what's going on based on where these elements show up on the chart. Why do we care about any of this? Well, biologists care about this because all of biology is driven by filling your outer shell. An outer shell means that you feel fulfilled. This hydrogen atom on the right-hand side has 1 electron in its outer shell, and it doesn't feel fulfilled. It's looking for a second electron, and there's a couple of ways that you can actually make that hydrogen feel fulfilled. You could add an electron to the outer shell. You could remove that electron and give it somewhere else. And there's multiple ways of doing that, and the same thing with carbon. It has 4 electrons in its outer shell, it wants 8. So putting 4 more electrons in the outer shell will make that carbon feel fulfilled. And we'll talk more about this as we go through this chemistry. So if you understand how many electrons you need to fill each of the 4 main elements of life, so hydrogen, carbon, nitrogen, and oxygen, what you see is that you need 1 electron to fill hydrogen. You need 2 electrons to fill oxygen. You need 3 electrons to fill nitrogen. And you need 4 electrons to fill carbon. And what this generally means is that that's the number of bonds that each of these atoms can make with other elements. So you can make a molecule called hydrogen, right? HH or H2. And the line that I drew here is a covalent bond. And that covalent bond allows for the sharing of electrons between 2 elements. And each of the hydrogens in hydrogen, the gas, H2, is fulfilled. With oxygen, you're going to do it. Oxygen uses 2 bonds, and so the easiest thing to draw here is water. So you see that oxygen has 2 bonds. So it's gained 2 electrons from the 2 hydrogen atoms it bonds with, so they're sharing those. And that allows for the formation of a very stable molecule, water. And that water is what drives all of life. Similarly, nitrogen will use 3 electrons from other atoms, and carbon uses 4. So the 2 things that we really need to understand are covalent bonds. And what are covalent bonds? Covalent bonds share electrons versus non-covalent bonds. Non-covalent bonds do weird things. So each of them does something a little bit different, some with electrons, some not with electrons. And we're going to talk about each of the 4 of these: ionic bonds, hydrogen bonding, van der Waals forces, and hydrophobic forces. And the picture that I have here for you is just to give you an idea, right? We're not talking about any one molecule being separate from others on cells. We have tons and tons and tons of molecules that are put together through covalent bonds, through non-covalent bonds, and they're all interacting with one another in this liquid soup of water. Not only that, but in the water you have a lot of ions, and all of this mixture is what drives the formation of the proteins and the carbohydrates, the nucleotides, and the lipids that drives the formation of life on Earth. So if you don't understand the chemistry, it's going to be really hard to understand all of the biology. So let's talk about what the subunits are. So if we're talking about subunits, let's just make this a little smaller, push it off to the side, because right off the bat, we are actually talking about elements, so hydrogen, carbon, oxygen, and nitrogen, and these get put together into something called molecules. And those molecules can be things such as sugars, amino acids, nucleotides, and lipids. I don't know why they don't have all 4 of them on here. All right, so these are attached to one another through covalent bonds to create molecules. Now, that's not the only thing that gets put together with covalent bonds. You can then have these amino acids, nucleotides, sugars be put together into something called macromolecules, and that can be done with covalent bonds and non-covalent bonds, and that creates, so from sugars, you get carbohydrates, carbs. From amino acids, you get proteins. From nucleotides, you get nucleic acids, and that's a string of nucleotides. And from lipids, you can make things like phospholipids and other higher ordered lipids. And interestingly then, these macromolecules then can also form together to make something called macromolecular assemblies. And what they're showing you here is a macromolecular assembly called the ribosome. It's made up of rRNAs and something called RNPs, ribonucleoproteins, and all of this together creates the structure that drives the function of biology. So think subunits, molecules, macromolecules, more macromolecules. So covalent bonds, right, I said share electrons. And sharing is very different than taking one or giving one away, yes? Sharing means you actually have to have some communication with the other person. So if you're sharing an ice cream cone with somebody, if you eat the whole thing, that's not sharing. So if you are sharing, you actually have to be in close, intimate contact. And so what they're showing you in this image, right, is two hydrogen atoms. And sometimes they're too close together. And so what happens is these nuclei, where the positive charges are, they get too close to one another. It's like being too close to somebody when you're talking, and they force one another away. If they're too far away from one another, the electrons can't interact with each other. So there's a distance that's just right for every single atom and how it covalently bonds to another atom. Okay, so hydrogen gas, right, apparently has this particular distance between the two nuclei, 0.074 nanometers, right? And you're certainly never going to know what those numbers are, but understand that it's a optimal distance, and that optimal distance is how it can share the electrons best without repelling one another based on the protons and neutrons in the nuclei of the atoms. So covalent bonds share the electrons. Covalent bonds store energy. The formation of covalent bonds requires energy, and that's based on delta G. Do you remember delta G? Page. Let's just open another page here and talk about delta G. Delta G is Gibbs free energy, and free energy is based on two things, right? One of those things is delta S, which is entropy, and the other is enthalpy, delta H. And notice this entropy is really what drives all of biology. Entropy is disorder. So everything will go to disorder if you don't put energy into it. So a delta S that is positive will have a negative delta G, and that means it is a spontaneous reaction. And that negative comes from this negative in the equation, right? So if it's a positive delta S, if this is positive, almost always that means that you're going to end up with a negative delta G. If you happen to have a negative delta S, that means you're going towards more order. Okay, you're cleaning up your room, order. All right, so if you have a negative, up here, what that does, a negative makes a positive. So the second part of this equation becomes positive, and delta G is a positive. And that means it's a non-spontaneous reaction. You have to put energy into it to make it happen. Okay, so what does that have with covalent bonds? Well, covalent bonds mean that you're taking, so here's a perfect example, you're taking two hydrogen atoms and you're taking an oxygen atom. So this is more disorder than when you create the molecule of water. So you have three elements here, and here you have one molecule. So delta S, this is a positive delta S, which means you have, I'm sorry, it's a negative delta S. Okay, so you're going towards more order. So the negative gives you a positive delta G, and the energy that's required to make these bonds is stored in those bonds. So no matter what kind of covalent bond, okay, covalent bonds, they store energy. We have two kinds of covalent bonds. You have non-polar, so this one is non-polar, the double bond between oxygen, because the electrons are shared in a way that's, the sharing is equal, versus polar covalent bonds. Polar covalent bonds means that you have one atom that sucks the electrons closer to it than to the other atoms. And oxygen is what I call a piggy woods, right? So it's a pig. It takes those electrons and makes them come closer, and that's due to its electronegativity. So oxygen is highly electronegative, and in fact, all of the atoms over on this side, oxygen, sulfur, chlorine, fluorine, they're all electronegative. Even nitrogen is a little electronegative, so they do not share equally, and that creates non-polar covalent bonds. Really, really, really important. I can't stress how important non-covalent. If you just look at water, remember I told you water is what drives all of biology. So water and oxygen. Oxygen has, so this means a slightly negative charge. So this is a negative charge on the oxygen because the electrons are closer to the oxygen. The shared electrons are closer to the oxygen compared to the hydrogens. But notice, each hydrogen, right, now is completely full. This oxygen, so oxygen needs 8 in its outer shell, it's full. The hydrogens need 2 in its outer shell, so it's full. Everybody's full because of the covalent bonding, but you get this partially positive charge on the hydrogens and the partially negative charge on the oxygen. And those positive and negatives are really important for how macromolecules interact with one another. Positives want to interact with negatives, negatives want to interact with positives. It just all works out. Everybody's happy. Slightly negative interacts with slightly positive interacts with slightly negative. So covalent bonds, I think, are pretty simple. It's sharing. Non-covalent bonds are a little more difficult because they do everything else. Just because two things aren't held together strongly by a covalent bond doesn't mean that they can't be held together strongly by non-covalent bonds. Any individual non-covalent bond, so one non-covalent bond, is not as strong as a covalent bond, but you end up with lots and lots of non-covalent bonds, so they make up for their lack of strength by having increases in numbers. And so non-covalent bonds mean that they can be transient. What does that mean? They can be on, they can be off, right? So they may only be around for a short amount of time. Some are stronger than others, and it's all about how many you have to generate a particular structure. So remember, the non-covalent bonds are ionic. Remember? So the next one is, there's 4. Okay, so you're going to have to remember, I know the last one is hydrophobic force. This one is van der Waals. And there is no order. This is just the order I remember them in. And so hydrogen bonding is number 2. Okay, it's just the order that I happen to remember them in. About ionic bonds. Ionic bonds are formed when one atom gives up an electron. And when you give up an electron, which is negatively charged, it makes that atom become positively charged. And the other atom gains an electron. Okay, so that atom now is negatively charged. And the classic example of this is sodium chloride. So you have an atom of sodium, which actually has 1 electron in its outer shell. 1 is a bad number in an outer shell. The first 3 shells want to be filled with 2, 8, or 8, all something you should already know. And so sodium looks at the chlorine atom and says, ooh, look, you're missing one electron that would fill your outer shell, so sodium gives its electron. Okay, so it gives its electron to chlorine, and now what you end up with is sodium has a positive charge. The chlorine atom becomes a chloride ion, and it has a negative charge, and everybody's outer shells are full. Notice on this side, the chloride ion outer shell is full, the sodium shell is full. There is still an attraction between the sodium and the chloride, and actually these form a lattice-like structure that makes the shape of cubes. So salt is bound together through these ionic bonds. It's not just that the electron is given off, all right? The electrons are given, but it creates this ionic bond. And so let me just play you this song and see if you can listen to the lyrics and understand what's going on with this song. I would normally be singing this to you. I could sing it here.
Just a little atom of chlorine, valence -1, swimming through the sea, making the sea just having fun. She's not worried about the shape or size of her outside shell. It's fun to ionize just a little atom of Cl with an unfinished shell. But somewhere in this sea lurks handsome sodium with enough electrons on his outside shell plus that extra one. Somewhere in this deep blue sea is a negative for my extra energy. Yes, somewhere in this form a positive find a hole. Then I suspected chlorine felt a magnetic pull. She looked down and her outside shell was full. Sodium cried, "Wanna guess be my bride, and I'll change your name from chlorine to chloride." Now the sea evaporates to make the clouds for the rain and the snow, leaving a chemical compounds in the absence of H2O. But the crystals that wash upon the shore are happy ones. So if you never thought before, think of the love that you eat when you salt your meat. Think of the love that you eat when you salt your meat.
So this song I heard many, many years ago by the McGarrigle sisters. They were Canadians, and I feel like one of them must have been a budding biologist at one point, or a chemist, to have written that song. Now, if you understand that song, you understand the love, the attraction between sodium and chloride. It doesn't go away. The salt crystals, right, in the absence of water, will form into a cube, and that cube is what we put on our meat. If you put salt in water, it dissolves. The sodium and the chloride ions can dissolve from one another. They become dissolved in solution, but if you once again remove the water from that, right, then you form the ionic crystals of sodium chloride. So the main thing I want you to get here is sharing of electrons. Ooh, right, sharing down here. That is based on covalent bonding, whereas when you give one and receive a different one, that is ionic bonding. All right, so we've done one of the 4 non-covalent bonds. Let's talk about van der Waals. Van der Waals attraction is all about how close two elements can be to one another. Remember when we had the hydrogen gas? Okay, we had hydrogen gas. If the nuclei get, right, if they're not close enough together, the electrons can't interact. If they're too close together, right, you had some repulsion, and that repulsion is based on van der Waals. So there is some optimal distance between electrons, no, not electrons, excuse me, there's an optimal distance between atoms where, right, the attraction and the repulsion are just right. So if you look here on this graph, right, if the distance between, so if you look, when the distance is too close, you have this repulsion, right, this is all being repulsed up here, until you get to just the right distance, that's here. And once you get past that distance, then the attraction is going to become less and less because you're too far apart. So there's an optimal distance where you get just the right interaction. That's based on van der Waals attraction. Hydrophobic force, right, the last of the four that I remember, right, we haven't talked about the second one, is all about right, what does hydrophobic mean? Hydro is water, so this is water. And what's phobic? Afraid of. So water-fearing molecules are nonpolar molecules. Nonpolar molecules means that they don't have any, so there are no partial positive or partial negative charge. There's no ions. Okay, so there's no charge. These are uncharged molecules. So nonpolar molecules like to stay together and stay away from water, whereas polar molecules, okay, polar molecules want to be with polar because they have these partial charges, and polar loves water. Okay, and that's because water is polar. So what is shown in this image? This image shows you some oil and water. The oil has all separated out from the water. If you were to mix this together, the oil would be in little droplets mixed with the water, but they will then glom together. They will aggregate. They will try to minimize their interaction, right, with water. Now they can't completely, they can't get rid of it unless they jump out of the glass, which we know doesn't happen. But by having just one surface where these oil molecules have to interact, that means that all of these molecules up here on top don't have to be interacting with water. So the minimization leads to particular structures. And you're going to see this is really, really important for how the first cells formed. It happened spontaneously because nonpolar molecules didn't want to have anything to do with water. Okay, so the last, right, of these, we're still talking about water loving and water hating. Okay, I hate the word hate, which is kind of a funny way to say that, but hating, let's just say afraid of. Okay, and that's what most things, when you're afraid of something, it's because you don't understand it, you're afraid of it. So hydrophilic means it likes water, and hydrophobic means it's afraid of water. And what's interesting is that you're going to see that you get lots and lots of macromolecules and molecules that can be both hydrophilic and hydrophobic at the same time. They can go both ways. And those molecules are called amphiphilic or amphipathic. All right, and so we're going to talk more about that, but in particular, specifically related with the cell, you have areas of the cell, like the membranes, that are hydrophobic, whereas you have the cytosol, which is hydrophilic. So there are things that stick through membranes that are both hydrophilic and hydrophobic portions on them. All right, well, so we did atoms, okay? We did atoms, and we talked about hydrogen, carbon, oxygen, and nitrogen. But we haven't really talked about the formation of amino acids, lipids, sugars, and nucleotides. Okay, and this is where I usually tell students, you're going to need to learn the amino acid structures, because I want you to know what the amino acids look like, I want you to know what lipids look like, I want you to know what sugars look like, and I want you to know what nucleotides look like. And that's going to be really important. So I think what I'm going to do is I'm going to stop this flip lecture here, and we'll pick up here when we get back together on Tuesday morning. All right, this is all going to be on a quiz, so prepare yourselves and have a good rest of your weekend.